Hello and welcome everyone to our organoid expert panel. My name is Jenna Mocha. I'm a senior product manager at STEMCELL Technologies, and I will be moderating the discussion during today's panel. Firstly, I want to introduce our panel members. First, we have Dr. John Stingle. He's our Associate Director with Research and Development. He's heading up the epithelial team. So Dr. Stingle oversees the development of epithelial cell culture products. We then have Dr. Ryan Conder. He is a senior scientist and team lead for the intestinal team, overseeing the development of products for intestinal organoid culture. Dr. Philip Kramer is a senior scientist on the intestinal team and the lead scientist for the newly released STEMDiff intestinal organoid kit for generating intestinal organoids from human pluripotent stem cells. And finally, Leon Chu is a scientist on the neural team. Leon leads the development of cerebral organoid products for deriving organoids from human pluripotent stem cells. So today's discussion will be centered around the questions you submitted to our panelists via our online form. If you'd like to submit additional questions, please use the Q&A panel or chat box on the right of your screen, and we will address as many as possible at the end of the discussion. So let's get things started, and we'll start things off with you, John. When it comes to nomenclature in the research community, there's a real lack of consistency in the terminology used for organoid culture systems. The word organoid is often thrown around and used for a lot of culture systems not classically considered organoids. And conversely, there are lots of different terms that people use to refer to organoids that are from different systems. For example, there's the organoid versus entoroid discussion. So I was hoping you could start us off by framing the discussion. When we're talking about organoids, what types of culture systems are we talking about?
Well, Jenna, you raise a very good point there. And I think the biggest confusion comes from organoids versus, say, spheroids. So spheroids evolved out of the neural field, particularly out of neurospheres. So for example, if you took a developing brain from a mouse and you dissociate it to single cells, and you put those cells into a culture dish where the cells cannot attach to the tissue culture plastic, most of the cells undergo apoptosis. But a subset of cells, the stem cells and early progenitor cells, can proliferate and form a neurosphere, a ball of cells. Now that ball of cells is enriched for other neural stem cells and progenitor cells, but it doesn't have an internal structure and organization that resembles anything you'd see in vivo, say in the normal brain. And of course, out of the neurosphere evolved, say, the mammosphere system. And again, in the mammosphere, you can take normal breast tissue, put it into a sphere assay. Most of the cells die, the stem cells survive and form a ball, mostly of basal cells. You don't get any other lineages in there. So now that's in distinction from an organoid. In an organoid system, you're taking a population of, say, a tissue. You can take, say, some mammary tissue, and you can dissociate it and put it into a culture system. But this time you're putting it in a system where the cells are embedded in a matrix. And typically we use Matrigel. Matthias Luthoff has described the use of PEG hydrogels in a Nature paper a couple years ago. But typically we use Matrigel, and the cells will proliferate and form a structure that has a certain degree of organization and it resembles the parental tissue from which it's derived. So if I'm putting, for example, a mammary cell into an organoid system, it's generating a layer of luminal cells, some basal cells. It has a central lumen. The epithelial cells can be polarized. And depending on the culture media that we use, we can have a certain degree of cellular differentiation.
Thank you. So Philip, there are several questions that were submitted about the value of organoids as tissue models. The characteristics of an organoid system in this context are obviously dependent on what type of organoid that we're talking about. So Philip, I was hoping that you could comment on the differences between adult stem cell derived and pluripotent stem cell derived organoid cultures.
Yes, absolutely, Jenna. So there has been reported differences in the field between adult stem cell or human biopsy derived organoids and HBSC or human pluripotent stem cell derived organoids. One of the major difference, for instance, in the intestine or in the lung system, is that the PSC derived organoids are more often fetal in nature. So Just as an example, for the intestine, they obviously they for instance lack the presence of a functional brush border enzymes or the presence of differentiated enteroendocrine cell subtypes. And in the lung system also, it has been reported that, for instance, the gene expression profile, if compared to a human adult sample, is not really matching the HBC-derived lung organoids. So what is the reason for that? Obviously, the medium we are currently using, they don't really enable the full maturation of HBC derived organoids to resemble the fully mature phenotype. Another example is also that, for instance, these in vitro biopsy derived organoids, they can further, the HBC derived organoids, they can be further matured, for instance, if they are engrafted in mouse models, and then they do resemble a more or near physiological characteristics, and are pretty similar to the more adult stem cell derived organoids. Then another difference between adult stem cell derived organoids and PSC derived organoids is, for example, that the PSC derived organoids, they are not always exclusively containing an epithelium cell type, but also, for instance, a mesenchymal cell type, which is also being present and described in the intestinal organoid system, if we are looking at PSC-derived intestinal organoids. Those mesenchyme is usually just a byproduct of the differentiation process and sticks around and provides important niche factor signaling to the intestinal epithelium and to the intestinal stem cells. But then also we have to think about what kind of model system is the best to choose if customers are evaluating organoid systems for their line of research. And therefore, we also have to consider the accessibility of samples. So for instance, if people are interested in studying disease models, then they may have not access to patient material from carrying inherited diseases. So therefore, it might be more accessible to use stem cells, which are and represent an unlimited cell source for their line of research, and then using genome editing tools like CRISPR-Cas9, TALENs, or zinc finger nucleases to derive their individual cell types.
Thanks so much. Yeah, it sounds like the context of the experiment is really going to determine what characteristics of the organoid system are going to be most crucial to weigh when determining what system you want to choose for a specific research application. So to go a little deeper on that into the topic of organoids as model systems, there are specific characteristics that give a model system characteristics that are well suited to modeling specific diseases, for example. So Ryan, can you comment on what makes a system a good choice for a particular disease model?
Sure, happy to. I guess that really speaks to the popularity of organoids and their usability, and I think that's one of the key things. As organoids are consistently challenged by researchers, they seem to step up to this challenge, and the results are really pushing the boundaries of what we know in research. For instance, in cancer biology, to grow a tumor organoid is very, very similar to growing a regular organoid. So this allows for the opportunity to take tumors from the various tissues and compare it alongside the normal material in vitro. And really nice publications have been shown for this, looking at colorectal cancers in the intestine, pancreatic, prostate. Recent publications have shown glioblastomas in the brain. So you're really able to dissect this disease a little more thoroughly. In other disease modeling systems, host microbe interactions have been looked at, both going back to the intestine. I guess enteric viruses, both noroviruses, rotaviruses, have been used in really nice co-culture systems. You can also look at bacterial pathogens. You can micro-inject directly into the lumen of the intestinal organoids and look at responses there. Throughout the brain organoids, really popular a couple of years ago was when the Zika virus had emerged. You were able to look at specific results in microcephalies in the brain. So really, really many applications. I guess one of the biggest ones too has been going back to the intestine, cystic fibrosis. A really nice assay was designed in collaboration with the Clevers and Beekman groups, where you could look at these organoids and find a very, very quick and simple assay through the treatment of the drug forskolin. You could see a swelling of these organoids. So you end up getting a very, very quick diagnostic for a specific disease. This goes back to the precision aspects of organoids, where you can look at individual patients and do drug screening and really find the appropriate treatment for the individual patient rather than a blanket approach. And I think this sort of speaks to how disease modeling will continue using organoids.
You touched on a couple of things that I'd like to go a little bit deeper into. Firstly, you mentioned using brain organoids for Zika virus infection modeling. Leon, can you comment on the specific advantages of cerebral organoids in that context?
Yeah, so in terms of using the cerebral organoids or most organoid systems, what's really key for them is they actually will contain different cell types. And these different cell types are impacted by the virus differently. So in the case of Zika virus, it's now been shown that the Zika virus seems to target the neural stem cell populations more than they do the neurons. And because of this, the cerebral organoid system is a good model to study this, because now you can study how Zika virus is depleting the neural stem cell population, how that affects the neuron population after that.
And as I understand, that model system also provided one where there wasn't a really viable alternative. For example, using mouse wasn't a really viable alternative in that application. Is that true?
Exactly, yeah. Species to species different. There are species differences in how the virus affects it, and the human one does seem to be impacted differently than the mouse models. So in that way, human-derived cerebral organoids were a better system to study this.
Awesome. Brian, you also touched on the question of cancer organoids, which is something that I'd like to address with John. So to move into this area where we had a real cluster of questions submitted around tumor-derived organoids. So John, can you first speak to the technical considerations? So what do researchers need to take into account when culturing cancer organoids versus those derived from healthy cells?
Okay. So probably the first and foremost thing that would be on my mind if I was trying to grow a tumor organoid, particularly from, say, a primary biopsy sample, or even, say, a metastatic sample to another part of the body, like this is the liver or bone, are the cells that I'm growing out actually tumor cells? So when we stick a needle, for example, into a prostate to biopsy a primary prostate tumor, that needle is going to capture some normal cells, surrounding normal cells, as well as tumor cells. And of course, a lot of organoid media are very robust media, and they can promote the growth of both types of cells. Likewise, it's been reported, particularly in prostate cancer, when trying to grow prostate organoids, is that if you go and sample a metastatic biopsy to the liver or to the bone, you could actually have normal hepatocytes or some kind of a bone-derived cell that will basically ultimately take over most of your cultures. So that's the first thing. We have to ensure that we are growing, the organoids that we're growing are tumor. Another consideration is just the difficulty of growing some types of tumors. Colon tumors have a pretty high success rate. Prostate tumors are quite low. There was a recent paper in Nature Communications where only, I think it was about 20% or so of tumors could be successfully propagated in organoid culture. The other considerations I would take in, if I was growing a tumor organoid, I want to first ensure that it has a morphology that does resemble the parental tumor, does express the appropriate biomarkers. So if I'm growing, say, an organoid from an ER-positive breast tumor, I want to make sure that my organoids also express the ER, estrogen receptor. And as well, would be good to confirm that the organoids have a genomic profile, mutational profile, that broadly resembles the parental tumor. And the data to date actually does look pretty good regarding this. There was a very nice paper from both Hans Klever's group, the Norman Sachs paper, as well as Nicola Valeri's group, where they looked at breast and GI tumors. And the mutational profile of the organoids actually did look pretty good compared to the parental tumors. And of course, the other real interest in tumor organoids in the field of cancer research is, can these organoids predict a patient's response to therapy? And again, the data on this is quite limited at this stage. But I do want to highlight the paper that was published in Science earlier this year by Nicola Valeri's lab from the ICR in London, where they looked at over 70 GI tumors and observed that they were an absolutely fantastic model for predicting patient response to therapy.
And that sort of leads into and anticipates some of the next question, which is how much information can be gathered from these systems in, the organoids growing in vitro, and what that means about the tumor in vivo. So you touched on some of the drug response characteristics of a given tumoroid and sort of the predictivity of that for in vivo drug treatment. Can you comment on how much information can be gathered from a tumor organoid on things like metastatic capacity or sort of the aggressiveness of a tumor or other characteristics that we might be able to learn from using the in vitro system?
Well, again, certainly for the metastatic point of view, it's a bit early days yet. Certainly we can draw on our experience from studying spheroids. We do know that in, say, breast cancer, very highly metastatic breast cancer cell lines generate spheroids that are very loosely packed, whereas less metastatic cell lines generate spheroids that are very compact. And if we extend that logic to an organoid, it remains to be seen if highly metastatic clones will generate organoids that are, say, highly branched or have a very different morphology from organoids that are very, say, spherical. And then with regards to other features, again, the main important thing is, say, for aggressiveness. If certain genetic mutations are identified with an aggressive primary tumor, metastatic tumor, are those mutations maintained in the organoid?
Ryan, maybe you can comment on some of the mechanisms that people have used to dissect tumor characteristics using organoids. So, for example, Dr. Sato's work looking at the media requirements of tumors as a way of more or less kind of phenotyping to infer the genotype of tumors.
Yeah, there's been a couple of approaches to really dig a little deeper into the tumors, and I think two approaches through the Clever's group and Dr. Sato's group were able to take the concept of how an organoid is grown with respect to the medium, whereas the specific pathways that are necessary to keep the stem cells in their active state and proliferative state was taken. So the key pathways that regulate this were individually removed through reagents in the medium as a screening mechanism. For instance, Wnt in the intestine is a key activator of intestinal stem cells. By pulling this aspect out, you're screening for tumors that specifically have mutations in the Wnt pathway, and in the intestine, APC is one of the biggest mutations. So really, really elegant experiments dissecting what mutation before the sequencing was even done on a global screening method.
Excellent. So moving now from...
sorry. I did have one more point I think I'd like to add to the cancer models. So just talking about also environmental aspect of looking at the cells in a pseudo-organoid model, in an organoid model in general, is that in the larger organoids, you also get a hypoxic core. And this type of environment too is also important environment for some types of cancer cells as well. So having that type of kind of gradient within the organoid also provides environmental niche for certain cancer cell types.
And that is an excellent segue into the next question, which is now moving from cancer organoids and into HPSC-derived organoids. We had some questions submitted specifically on techniques that can be used to manage that kind of cell death in the interior of larger organoids, such as those cerebral organoids generated using the Noble protocol, for example. Mm-hmm. So Leon, can you address any sort of techniques that can be used to mitigate the effect of that kind of hypoxia in the organoid core?
Yeah, so the primary effect that we're trying to improve here is media or nutrient penetration into the center of these organoids. So this has been addressed using a few different methods. Currently, the ones that are being used are environmental factors. So in the original publication by Jürgen Noblick and Madeline Lancaster, they tried to use orbital shakers or bioreactors as an environmental method to basically create more nutrient diffusion into the center of these organoids. Additionally, labs have also been using high hyperoxic conditions, so high O2 at 40%, to also kind of allow better oxygenation into the center of these organoids. And also there have been groups, mostly from Yoshiki Sasai's group, that have even tried passaging organoids through cutting the organoids. So they don't actually dissociate them, but they'll actually physically, mechanically use some scissors to break the organoids apart. And they can continue to grow those guys for longer term. Some alternative approaches that are now starting to come online, but I don't think we have really well-defined methods yet, are trying to introduce vasculature into the organoids using co-culture systems using endothelial cells. So groups like Ben Waldau's group has used endothelial cells mixed in with cerebral organoids, and they found that they could actually form capillaries within the organoids spontaneously. Additionally, they've also attempted transplantation into a mouse, and they've also shown that capillaries could invade the organoid and create channels in that way. Finally, I think there's another method that is also being studied, are bioengineering approaches. So somehow introducing pores or channels into organoids artificially, I think can also be a method that can be explored to help with this.
Fantastic. So keeping then on the topics of culture techniques, we had several questions about supplementary techniques for applying organoid cultures to specific research questions. And specifically, there were inquiries about monolayer culture systems as well as co-cultures with either bacteria or immune cells seem to be sort of the most popular directions that people are trying to go with these culture systems. And also specifically tools that make these sort of supplementary techniques or ways to use the cells generated from organoids as user-friendly and accessible as possible to the general research community. So Ryan, can you comment on using techniques like monolayers or co-cultures?
Sure. I guess this is what we're watching is the evolution of organoid culture right here. Whereas the first wave of publications and discoveries that came out were how to recapitulate a stem cell niche and how to grow a specific organoid from a specific tissue. And this was an incredible feat, and the amount of tissues that we can now grow and use in vitro is fantastic. But the next step is looking at the functionality and what assays we can do, what we can really learn and mimic these human model systems. And I think some of the ones you mentioned, specifically a monolayer culture to start, I guess has been an industry standard for a long time, using immortalized cell lines. So you could take a tumor line, basically grow it in a monolayer, either on feeder cells to support the signals, or with a media that supports the growth of these, and able to ask questions specific to, for instance, in the intestine, barrier function, permeability. And this was great. We've learned a lot. Many drugs were discovered based on this protocol. But what really lacked was the physiological relevance and as you're looking at tumor models. And I think organoids have provided a step where you can take a patient biopsy really amplify the stem cell population and look at a monolayer that is now specific to the patient. And this was never possible before organoid cultures. So early studies looking at these have demonstrated you can use an air-liquid interface and get apical access to this epithelium. You can go basal access and recapitulate the luminal and external portion of the intestine. Bacterial studies have been done. Early studies looking at immune response have been possible on these. And really moving forward, this could be the step that takes organoid culture into a more high throughput aspect for drug screening.
Can you comment on how approachable that technique is for a lab that might be interested in starting to generate monolayers from organoids?
Yes, it's incredibly approachable. The system is very straightforward. Taking the organoid cultures that are grown in matrigel domes, simply dissociating these into a monolayer on a layer of matrigel and adding the media within a week gives you a very confluent monolayer, which has been shown to have clear barrier function, ion channel response, and amenable to all these applications.
And with respect to co-culture systems, am I correct in my assumption that once the cultures are generated, they can definitely withstand sort of short-term exposure to other culture conditions? And so, if you have cells that are very sensitive to the media that they might be in, that you want to expose to the organoid cultures, that can be swapped out for the organoid media for that interaction period and that kind of thing, so that those systems also help facilitate these co-culture conditions?
Yeah, you are correct on this, and it's still very early in some of these publications. I think the concept of co-culturing identifies one of the biggest shortcomings of the organoid field. You're isolating a specific tissue. What we're able to do now, and it seems that this is supported endogenously, is add these different tissues back in, as Leon was getting at earlier, talking about a vasculature and endothelial cells. This has been demonstrated. One of the nicest publications I've looked at or I remember on these is co-culture with neural crest cells, looking at in the intestine, where the Wells lab was able to demonstrate enteric nervous system function on the intestinal tract and actually see the pulsating of this tissue. So it does seem that the current media that's being used does support these alternative co-cultures. When we finally move towards a more complex system where we are getting the endothelial cells, creating a vasculature, creating nervous system response, looking at immune systems, there probably will be modifications, but this is really the advancement of the field.
You actually touched on something there that was one of the questions that's submitted from our audience here, which is how can organoids overcome the limitations of the lack of vasculature and also recapitulation of organ-organ connectivity. Philip, is that something that you might be able to address?
Yeah, so I think there we certainly look into co-culture models where we really add endothelium cells into an existing in an existing organoid system. We have also, I think, in the field, especially from PSC-derived organoids, it has been shown that, for instance, the additional VEGF in the kidney differentiation system has the ability to enrich for endothelium cells. So you have possibilities to add those vasculature into the system. Then, of course, furthermore, what Leon also touched base already early on is in an in vivo engraftment into mice. Then the vasculature, which is normally derived from the organoid, as long as they have progenitors or endothelium cells, is being able to further mature into a more prominent vasculature. If there are no progenitors present, then the mouse system or the host is actually able to generate a vasculature.
Fantastic. And continuing with you, Philip, while techniques for growing organoids are starting to become a little bit more standardized, although there's lots of work to be done in the field there, methods for using these cultures to generate quantitative data are still kind of in the process of being standardized, and for the most part tend to be left up to the individual lab level to decide what they're going to measure, how they're going to measure, but there doesn't appear to be a lot of standardization for those metrics across the literature. Can you comment at all on methods for quantification of results in organoid cultures, and specifically, you know, if you're a lab looking at starting to use these cultures, what kind of data in a quantitative fashion are you really going to be able to get out? What can you measure?
Yeah, Jenna, you're absolutely right that many quantitative assay have been developed for multiple organoid systems in a variety of contexts. So just to mention, I think the most common ones we are using for our medium development in our organoid systems are really very simple ones. For instance, live or time-lapse imaging, combining those with image analysis software to capture and quantify the growth of our organoids and comparing different media conditions. Other quantitative assays have been developed and used, for instance, in combination with certain drug screenings, for instance, looking at the toxicity of those drugs and by measuring the viability, looking at EDU-based cell proliferation assays, but then also other labs, for instance, Friedman and colleagues have published the drug screening by using cisplatin on the kidney organoids, on HBSC-derived kidney organoids, and using an ELISA for a biomarker to detect kidney injury in this organoid system. And the biomarker was, for instance, KIM1. It's a kidney injury molecule. That is one way to actually correlate the effect of a drug on the kidney organoids and do this in a quantitative way. Then we have already heard earlier from Ryan that Deckers et al., they have published and very nicely developed a quantitative assay called the phospholins-induced swelling assay for testing cystic fibrosis drugs in patient-derived adult intestinal organoids. And then other groups, they also use quantitative uptake assays by using a deep fluorescence labeled deep peptide, or even measuring, for instance, the pH in the lumen of fundic-like gastric organoids. So all of these different methods just show you a variety of quantitative assays, depending always on the context and the organoid cell type.
It seems to speak a lot to the flexibility of various organoid culture systems to be able to interrogate the research question at hand. I'm going to add one thing to that I see a lot in the literature, which is the use of just the ability to form organoids as a metric for stem cell activity or stem cell functionality. It occurs to me that there might be somewhat different metrics for some of these cerebral organoids. Leon, do you have anything to add to Philip's comments?
Yeah, I mean, in terms of functional aspects, when you're looking at neural cells, the electrical activity of neurons is an important one. So we've been shown many times that cerebral organoids are, the neurons in them do form networks and they are electrically active. So some measures of either patch clamping, calcium signaling, or even just using multi electrode arrays, so MEAs, and measuring electrical activity of the cerebral organoids in the absence or presence of certain drugs as well can give you a quantitative readout of how effective some kind of screening is going.
So it seems to, again, circle back to the specificity of a given organoid system for the biology of the underlying organ that you're modeling with it. And so the readouts being very specific to the function of that organ as far as what's going to give you sort of the most informative readout. So one of the reasons that quantification has been a little bit of a challenge in the field has been something that continually comes up over and over again in conference proceedings, review articles, and again in questions submitted to this panel, which is the variability of organoid cultures. So Leon, can you comment on any practical strategies for mitigating variability in organoid cultures?
Yeah, so there's a few things that we can kind of address here, but I think one of them are actually just looking right at the media requirements of the organoid systems. So in terms of the traditional methods to derive organoids, in a lot of cases, they grow them in undefined conditions, and this includes stuff like fetal bovine serum, and even using matrigel, which is a very complex mixture of different components. So really trying to understand what are the underlying components within these undefined reagents that will allow the organoids to grow and form is pretty critical. So really elimination of these undefined reagents and standardization as well is one method to really kind of help this out. And I think John's got a comment here.
Yeah, so my team, when we were developing mammary and prostate organoid media, variability between experiments and even within experiment was an ongoing problem. And one thing we realized that really helped to reduce variability is actually when we dissociate our cells, or say if we're seeding an organoid culture or passaging an organoid culture, we found we could really reduce the variability just by dissociating the single cells and seeding as single cells and passaging as single cells. And because we found as clumps, it's very hard to get a very uniform size of a fragment of an organoid when passaging. And doing a single cells, it really reduces the variability, although it may be challenging to maintain cultures for an extended period of time when passaging as single cells because you lose cell-cell interactions where you need to maintain a niche. And passaging as clumps is better for maintaining a culture for long term. But if you want to do a quantitative analysis, my advice would be to seed as single cells.
Yeah, so technique-wise, definitely somehow controlling it and making passaging it definitely would be a benefit there. I can also speak at another kind of very important thing that might be overlooked by a lot of labs, and I'm talking about from the HPSC perspective, is inherent cell line variability, but also cell quality control. So one of the most important things for HPSC differentiation is really making sure you have very high quality human pluripotent stem cells, and you really need to check for very specific parameters. This includes like karyotype, undifferentiated PSC markers, morphology, and even when routinely testing maybe trilineage potential as well, just to make sure that the cells you're making are truly pluripotent. So cell quality control, I think, is something that a lot of times is overlooked, but making sure you're really keeping on top of it and checking to see that your HPSCs are truly pluripotent is so very important for any kind of differentiation protocol.
I know I'd even extend that to if you're maintaining organoid lines for extended periods of time in the lab, not even PSC lines, but adult tissue stem cells as well. I'd emphasize regular cell checks, especially karyotype.
For many of the organoid culture systems, especially those on the epithelial side that have sprung from the technology developed from the Cleavers lab originally, then the media composition also tends to be very complicated and also involve undefined components such as conditioned media. And I imagine that can also contribute to a lot of variability both within a given lab, between sets of experiments, as well as across labs for collaboration and that kind of thing. Ryan, I know you've had discussions with some key labs involved in multi-institute collaborations. Can you comment on the impact of organoid culture variability on collaboration and reproducibility?
Yeah, I can, and it is quite a challenge. I think you would have got from that last question everybody's comments on it, talking about the variability not just between labs but between experiments. So this is a real issue. And Jenna, as you pointed out, this is this is really state of the art right now. What has been discovered in terms of media composition, which is necessary to maintain not only the stem cell populations, but in many cases the differentiated cells as well, has been quite a challenge. And each lab works with their various media, their technicians, to come up with what works best in their lab. And this makes sense, and this historically is how it's been done for quite some time. And I guess when you're getting into this advancement in science, where you're taking these labs that have so much expertise in the specific questions they address, and getting together and forming collaborations to really address bigger questions or diseases on a whole, this variability becomes compounded. You're really looking to compare data discovered between labs, and if it's not standardized, if you don't have a method really to eliminate the aspect that one lab may be different than another's strictly on culture conditions, this is a pretty big issue. There's multiple ways to solve it. I guess just communication between the labs and standardizing is very, very key. Reagents, what is used, probably is one of the largest determinants. If you can get away from conditioned approaches within individual labs, this is an incredible source of variability. As John was commenting on too, techniques, how you're actually culturing these organoids between lab to lab has incredible differences. Really breaking it down to what is used, where you're getting the specific reagents, and the protocols used are probably very, very important in standardizing this.
Thank you. So we've gone through a lot of the themes that were submitted during the via our web form, and we've had several questions that are coming in live as well. One of them really ties in nicely with this theme of culture variability and surrounds the use of Matrigel, which is, there have been a lot of recognition that Matrigel can be an issue in these culture systems, in that it does have some inherent variability. We'd love to address the idea of alternatives and work in the field that's being done in this area. John, you touched on this with respect to Matthias Luthoff a little bit earlier. Would you like to start off the discussion on Matrigel?
Sure. So one comment about Matrigel is my group has had problems using it, and it depends on the concentration of the protein in the Matrigel. You have to make sure you get a Matrigel with a minimum amount of protein. I'm sorry, I can't remember the number off the top of my head, but it probably depends on the application. But we find that sometimes our organoids will sink to the bottom of the dish, and then they grow up as a monolayer underneath the dish. And we find using Matrigel to try and identify lots that are a higher protein concentration can solve this. As for, defined gels, a lot of people around the world are very interested in this, and many people are working on this. It's been a challenge to date. The best I've seen so far is from Matthias Luthoff's lab. He had a paper in Nature, I think about 2 years ago, where he used PEG hydrogels, and he added in different components in that, and was able to demonstrate he can grow mouse intestinal cells. But I haven't seen anything yet applied to more broadly to more tissue types. I'm not sure, Ryan, if you have anything you want to add to that.
No, I guess I can just agree. Matrigel seems to be a necessity we all use, and there have been lots of work to get away from this. And I think it's accepted that we would all like to get away from an undefined component, which is necessary to recapitulate the extracellular matrix. I guess one thing that would go into this is experimental considerations, right? Matrigel is absolutely necessary for the long-term passaging of these organoid cultures, but you really need to break down what type of experiment you're doing. And often you can get away from a Matrigel and go into a more defined component, such as hydrogels, really for limited passages after. And if these are the key experiments, the functional aspects you're looking for, a lot of these defined matrigels may be convenient. I would say a major caution here is to understand what's happening at the cell fate level. If you're changing off matrigel and not supporting a stem cell population, your organoids will survive for a couple weeks or a couple passages, but what's happening at the physiological level may be unknown. You could be losing an entire stem cell population and really assaying just the differentiated cells. So I think real critical checks of what your starting population would be and a final population would be essential.
Yeah, some of the work has been done by Andres Garcia and Jason Spence's lab, where they have demonstrated that a defined extracellular matrix protein composition was able to grow PSC-derived intestinal organoids for multiple passages in vitro, and that is really, I think, now holds a great promise to get away from these mouse sarcoma derived matrigel and enabling clinical translation towards tissue engineering and regenerative medicine.
Thanks. That's actually an excellent segue into another one of the questions that we've been asked, which is looking at a prediction around the table of how we think organoids can be used in preclinical stages, not just in basic research, and also maybe in clinical applications as well down the road, and sort of what the path looks like there, what types of applications might be a little bit more near-term versus far-term applications. Leon, can you start us off?
Yeah, so I think the organoid model cannot be viewed as this intermediate model between this more simpler kind of 2D monolayer models, and then before you go into these animal models. So that's how I've kind of pictured or envisioned where the organoid model kind of sits in this kind of timeline for preclinical screens, and basically. Because the organoid model is can contain many different cell types and it's more complex, I think that's a good kind of position that you can think about it.
Excellent. Philip, can you comment on how you see the application of these systems in a more translational or clinical context?
Yes, absolutely. So I think everybody agrees that these organoids, they hold great promise. And me from the PSC derived organoid side, I still believe we are very early to generate any PSC-derived organoid tissues which are or which can be used and utilized in clinical settings, because there's still a major concern with genetic stability in those organoids. Plus, controlling that seems to be feasible, but not potentially very fast and also very costly. So whole genome sequencing or exome sequencing would be one way to confirm that non-detrimental mutations has been acquired in vitro, which then will be translated into a regenerative medicine approach. So that would be my major concern.
Brian, can you comment on the same question?
Sure. I think the comments we just heard really set the stage for it, talking about this promise of organoid cultures. We've always had the promise of stem cells and the terms precision and personalized medicine thrown around. I think we do have a bridging of a gap here, taking from very primary research, which is necessary, to go translational, and some early experiments and early publications. A key one from the Watanabe lab in Japan really showed, in collaboration with the Clevers group, that these transplantations, at least taking intestinal organoids and going into a host mouse, you had a beautiful incorporation of the epithelia into the host's intestine. Junction formation was very fluid. Leon was commenting about activation of endogenous host systems on this. And really the promise is there, where you can take an organoid. We know we can do genetic editing on these. We can fix possible mutations and go back into hosts. And there are some enormous amount of cautions and ethical concerns that Philip was getting at, that I think we have the tools, or the tools exist, to really work through these and get to the point where we are treating individual patients rather than an individual disease.
And to close us out, John, can you comment on some of the preclinical and clinical applications you see?
So yeah, again, I think in the field of cancer, organoids are very exciting. Again, can they predict a patient response to therapy? I mean, again, it's early days right now. I mean, the papers are just coming out, and there's going to be quite a body of data required to get this into the clinic. So I don't see it coming in the next few years, but who knows, maybe a decade down the line, it could be part of routine clinical practice. But I think to get there, we're going to need a couple things. First of all, we're going to need standardization of these assays. Consistent formulations that are standardized from groups, so it can all, there can be collaboration, sharing data, and as well standardization of these workflows. But also we need to really be able to grow some of these difficult to grow tumors. When you have some of these slower growing, say a prostate tumor or a breast tumor that take years to develop, how can we generate an organoid of these cells in the time to do some drug testing and still treat a patient? So, but those are the challenges for the years ahead, and hopefully we'll get there.
Thank you. I think that's all the time that we have for our panel today. So I'd like to thank everyone for attending, and I'd especially like to thank our panelists for the discussion. A recorded version of the panel will be available on our website soon, and we will send all of the registrants a link as soon as possible. We will also be putting up some material answering some of the questions that we weren't able to get to, as well as listing some of the resources and publications that we mentioned during today's session up alongside that recording, so that if you're interested, you'll be able to reference some of the materials that we've been discussing. Thank you all again so much. It's definitely an exciting research field to be a part of, and we look forward to seeing where things take us in the next several years.